this whole problem is not without ethical issues involved. And why is there so much controversy over the issue of human embryonic stem cells, and specifically human embryonic stem cell research? So, on the one hand, you could argue that human embryonic stem cells are typically cultured from fertilized eggs. On the other hand, though, you could argue that those fertilized eggs, in fact, entail the destruction of a fetus when you then culture them to produce embryonic stem cells. But you could argue and say, well, human blastocysts and embryonic stem cells can be created with egg and sperm and tissue culture. As I've just described to you, there is no womb that's necessary in order to take embryonic stem cells and culture them to the point of blastocysts. And so, in fact, you can take a human egg and combine it with sperm in a petri dish and be able to generate blastocysts from which you can then culture embryonic stem cells, as I've just described to you. This is the same technology that's already in place across the world in various different advanced medical institutions that conduct in vitro fertilization to generate babies for parents that aren't able to generate babies in the normal fashion. So embryos from in vitro fertilization are often discarded or kept frozen and unused. Isn't it important then that we take these unused or discarded embryos from in vitro fertilization and make good use of them in research? Many researchers feel that that should be possible, but then others believe that we could take these embryos and instead offer them to another woman who would like to have a child. So these embryos could still be used instead of discarded, could be used by others to carry them to term in a woman. But, as I said, we could use them for research to learn how to generate neurons, pancreatic islet cells, muscle cells, for treating various different human disorders and injuries for which we have presently no cure. So what are the potential ways in which we might alleviate these ethical concerns? Well, scientists only recently have learned that it's possible to clone embryonic stem cells by taking an 8-cell embryo in a petri dish from a blastocyst produced by in vitro fertilization and just remove one single cell from that 8-cell embryo, and those 7 cells from the remaining embryo can still generate a normal human being by in vitro fertilization. Hence, no human lives are going to be lost in this process because these 7-cell embryos can still generate a normal human being. But we could take that one cell and expand that cell, as I've just described in tissue culture, to generate lots and lots of embryonic stem cells. So maybe we could get our cake and eat it too using this particular process. The technology, however, can also be used for couples who have a high risk of passing on a serious genetic disease and who wish to combine genetic identification of embryos with in vitro fertilization to produce a healthy baby. So in fact, this type of technology is already in use in various different clinics where a couple does have a severe risk of passing on some very severe genetic disease to their offspring. And rather than to do that, they would rather have their embryos screened to make sure that they can identify which one is bad and which one is good. And they do that by this type of technology, removing one cell, taking the other 7 cells, testing that one cell, and finding out is it good or is it bad, and then taking that 7-cell embryo and producing a child. The problem is that, in fact, there are some risks to the embryo by using this type of technology. And so there are many who believe that the risk for parents who might otherwise pass on a very serious disease, perhaps life-threatening or psychologically devastating disease to an offspring, that risk is worth the risk, because otherwise there's a huge risk, a 50% risk, that that child might be born with some terrible disease. But with regards to research, one obviously would not want to take those 7 cell embryos, considering that risk, and actually implant them and create a normal human being. There's too much risk involved for that sake. So for some, and in fact for many, this is really not an ethically acceptable way in which we could alleviate the concerns that do exist for those who believe that destroying the embryo, even though we can generate that embryo in vitro in a petri dish, is still problematic.